السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. اليوم أو الآن بشرح synapse and the neural integration. So I'm going to define the synaptic transmission. كيف السينابس ينتقل من مكان لمكان، ونعطي أنواع حق السينابس، الميكانيزم حق الأكشن potential الميكانيزم of action of the chemical and the electrical synapse. طبعا هذا التايب we're going to go over it all إن شاء الله. Synapses and the neuronal integration. So أنا أبغاكم تركزون وتتخيلون معي. النيورون اللي هو basic unit of a nervous system is the neuron. Nervous cell, it's the neuron. It can terminate, تنتهي on one of three structures. يا إنها تنتهي على المصل، هذا النيورون اللي أنا قاعد أتكلم عنه، يا إنه بينتهي على مصل، هذا ممكن يكون مصل, or it could be a gland or another neuron. So if this neuron I'm talking about is terminating on another neuron, we call it a synapse. This thing called a synapse. If the neuron I'm talking about is ending or terminating on a muscle, we call it a neuromuscular junction. Neuromuscular junction. We want to focus on the neuron terminating on another neuron, the synapse. The synapse is when a neuron junction between two neurons. It's called the synapse. So put this in your mind. Which one is the synapse? One neuron and another neuron. Right. Here we want to put the fundamental stuff and the basics of this lecture in this slide before we move on. So this is very important. So أبغى أتكلم عن النيورون الأساسية هذه. The first neuron. Focus. This is the first neuron. طبعا هنا راح يكون عندي cell body, dendrites, whatever. This is the axon. This is the termination of this neuron. So the first neuron, I'm going to call it the presynaptic neuron. The second neuron synapsing on it, it's called the second neuron here. It's the postsynaptic neuron. So this is the presynaptic, قبل ما يكون synapse. The postsynaptic neuron is the second neuron. Okay, so the action potential actually travels through the axon. طبعا أكيد generated through the axon hillock زي ما تناقشنا بالlecture اللي قبل. It's going to travel through the axon, and when it reaches the axon terminal, this part of the first neuron, it's going to open the calcium channels. It's going to open what? The calcium channels. This action potential, it's going to open the calcium channels, letting the calcium go in. And these vesicles, the purple vesicles, الموف, هذول vesicles contains a neurotransmitter. The orange one is the neurotransmitter. So when this vesicle is opening on the synaptic cleft, it releases the neurotransmitter. Whatever kind of neurotransmitter, so we have more than one. بنتكلم عنها بالسلايد الجاي. Later on, we're going to discuss the type of neurotransmitters. So just imagine any type of neurotransmitter, whether it was norepinephrine, epinephrine, dopamine, whatever. This orange dot is representing a neurotransmitter. So when this vesicle releases the neurotransmitter into the synaptic cleft or the synaptic space, this is a very teeny tiny space between the two neurons, the presynaptic and the postsynaptic. So when the neurotransmitter is released, whatever the neurotransmitter is, these we're gonna have, like for example, let's talk about if these neurotransmitters, all the orange dots are acetylcholine. put that in your mind. It's very common. So acetylcholine over here, whatever neurotransmitter we're gonna have on the end of the presynaptic nerve, it's always gonna have an antidote أو شيء ضده أو عكسه. Enzyme that's gonna destroy the acetylcholine. So أنا عندي هنا النقاط الأورنج البرتقالية acetylcholine طلعت من هذه الفيسيكلز into this synaptic cleft. عندي الآن النقاط الوردية أو pink dots representing the enzyme or the antidote opposing to the acetylcholine. It's called the acetylcholinesterase. If we have A's at the end, it means it's an enzyme. Basically, it's gonna destroy the acetylcholine and it's gonna break it down. Later on, these fragments of acetylcholine, it's gonna be uptaken and recycled into this neuron, and it's like this cycle. So we have acetylcholinesterase. So now I told you about what happens if an action potential comes over and we have a calcium channels opening, and then the vesicles release their acetylcholine, and then we have also the acetylcholinesterase here acting in the ECF, acting as an antidote. It's gonna break down the acetylcholine, so we don't have a buildup of acetylcholine here. It's gonna break it down, recycle it. But the most important thing is that when acetylcholine here, before it goes and gets broken down, what does it do? When acetylcholine gets released, it actually goes to the acetylcholine receptor. Acetylcholine receptor on this postsynaptic, the second neuron, the postsynaptic neuron, the second one. This is our aim. Why did I release the acetylcholine from here? So it can go and bind with the acetylcholine receptors. And we're gonna have graded potential. As we learned in the previous lecture, if we have more than one graded potential affecting on the same nerve, it can be summed up, so we can add them together. And if we add them together and the result was actually above the threshold, then this graded potential will move on into an action potential. So this is basically the summary and the sum up of this lecture, but we have to go actually and furthermore to discuss the details. So I just want you to think about this diagram or drawing, whatever, when you study, and imagine things happening, because it all makes sense. It's like a story. So why do we study this? It's very important in medicine because later on طبعا هنا المهم عندي acetylcholine، لأنه ليش أنا أصلا أجريت الأكشن بوتنشل؟ عشان نطلع الاستالكولين الموجودة بالفيسيكلز هنا وتطلع بالسينابتيك كليفت، بعدين الاستالكولين تروح ترتبط بالسكند نيورون وتسبب لي graded potential. So إذا كان عندي patient أنا for whatever reason there is, I want to increase their acetylcholine، أنا راح أعطيه دواء يمثل إنه هو acetylcholine أو يسوي نفس acetylcholine، نسميه a medication that mimics acetylcholine function. It's gonna mimic the acetylcholine, but what's the difference? It maybe stays there for a while longer than acetylcholine does, because the acetylcholinesterase may not work on this medication I'm giving the patient as well or as efficient as it does with acetylcholine. So this is the basic clinical application. Okay, now let's go through the lecture. Synapses. Synapses قلنا between one nerve and another nerve. Junction between two presynaptic and the postsynaptic neuron. طبعا احنا ما نقول one nerve and another nerve. نقدر نقول إن a presynaptic nerve and the postsynaptic nerve, two nerves يعني. So we have two types of synapse. I want you to focus here. The two types of synapse, it's very important because the electrical synapse, it's rare. We don't use it as much as we use the chemical synapse. Chemical synapse, I want you to put a very important sign next to it. Why? Chemical synapse. because the chemical messenger is transmitted across the junction separating the two neurons. It's basically where the neurotransmitters are released. It's an indirect communication, but that's actually what happens in humans. So it's important because it's 99.99% of the time it's chemical synapse. It's rarely an electrical synapse. Electrical synapse, it's rare, but it's also there. It's direct communication. It's less common, but we're gonna know the examples. So basically, electrical synapse happens through gap junctions. This is one of the most important informations about electrical synapse. It's rare, happens through gap junctions, and plus the examples. The pulp of the tooth and the retina of the eye, cardiac muscle, and smooth muscle. So basically, it's touching upon this topic very superficial in this lecture. بس يعني ما يحتاج إننا نتعمق فيه. Just note 1 or 2 examples. It's enough, as the doctor said. يعني. Yeah. We're gonna focus on the chemical synapse because it's the most common in human beings. In human, the CNS, the most of these synapses are chemical. It's always chemical, most of the time. So information from one neuron to another goes through chemical synapse. Now let's talk about the synapse. So this is a cell body of a neuron. This is another neuron synapsing on this neuron. So this is a pre-synapse. It's sending this message to this. This is the receiving end. This is the sending end. And we can call this a presynaptic because it's sending its information. This one is a postsynaptic cell body of a postsynaptic because it's receiving the information, the action potential actually. The synapse. Let's talk about the synapse. This is the same as we discussed. This is the presynaptic axon. We have the action potential traveling through this axon until it reaches the terminal, opens up the calcium channels. Third, calcium goes in. When the calcium goes in, the synaptic vesicles containing acetylcholine or whatever neurotransmitters we have more than one, not only acetylcholine. We can have norepinephrine, epinephrine, dopamine, whatever. For now, we'll just stick with the example acetylcholine. So when the calcium goes in, the vesicles go and diffuse and release their neurotransmitters into the synaptic cleft or the synaptic space. The acetylcholine will go into the acetylcholine or the neurotransmitter receptor, go inside and generate a graded potential. So we're going to have more than one graded potential, sum it all up, and then we have an action potential. So now let's talk about the properties of this synapse. So this synapse, the first thing we're going to know is very important clinically. It takes time. It's called the synaptic delay. أول خاصية من خصائص السينابس, it's synaptic delay, the transmission of the electrical impulse of the action potential, because you're gonna have the action potential traveling on the presynaptic. بعدين الكالسيوم تفتح، الكالسيوم يدخل يعطي إشارة للفاسيكلز إنها تروح تسوي ريليسنج للنيوروترانسميتر. هذا ياخذ وقت لين ما إنه يسوي graded potential. بعدين نجمع graded potential ويكون action potential. So for an action potential to actually happen, it's gonna take time from 0.5 to 1 millisecond. So make sure it's 0.5 to 1 milli, not even second, millisecond. It's a synaptic delay. The definition is that for the action potential to generate by chemical means from the presynaptic neuron to the postsynaptic neuron takes time, basically. So easy. هنا نركز شوي. So the postsynaptic potential, it's very important. So we have synapsing. Signal at synapse either excites or inhibits إنها تحفز أو تضبط the postsynaptic neuron. So two types, the excitatory synapse and the inhibitory synapse. Let's focus here on the diagram. So if my graded potential was less than the threshold, طبعا الخط الأخضر هذا هو threshold تمام. If my action potential depolarizes just a little bit, not a lot. Depolarizes a little bit. يعني خليه less negative. يعني كان minus 70 صار minus 50. This is an excitatory synapse. Basically less negative means excitatory. كأني أبي أحاول أسوي action potential بس ما صار. يعني it didn't get through the threshold potential, but it's an excitatory. But here it's like I'm hyperpolarizing because I'm increasing the negativity. I'm hyperpolarizing, so the threshold is going further away from the threshold. It's inhibitory synapse. So the excitatory synapse actually depolarizing, good for the action potential generating. So if I had another EPSP and another EPSP and we sum it all up, we could have an action potential at the end. But here, IPSP, if I have two IPSP, it will never get to an action potential. It's just going to go further down away from the threshold. So this is basically the idea. As I said, neurotransmitters, most important one, acetylcholine, dopamine, norepinephrine, epinephrine, glutamate, and gamma aminobutyric acid or GABA. So vary from synapse to synapse, the type of neurotransmitter. Some neurotransmitter is always released at the particular synapse. So ممكن يعني زي ما درسنا بالأنات إنه acetylcholine is always released from the preganglionic سواء كان بالsympathetic أو parasympathetic. And the parasympathetic postganglionic دائما acetylcholine. النورابinephrine دائما كان بالsympathetic postganglionic. So مو موجودة بالlecture, just for a reminder. Quickly removed from the synaptic cleft. يعني لو كان موجود بالسينابتيك كليفت الأستالكولين قلنا بيجي الإنزيم اللي هو اسمه acetylcholinesterase يكسر الأستالكولين يسوي له الريسايكليشن عشان ما ينفع يقعد هناك وقت طويل بالECF extracellular fluid. Important, glutamate is the most common excitatory neurotransmitter in the brain. You just have to memorize this because excitatory, of course, it's going to be EPSP, which is excitatory post-synaptic potential. Post-synaptic potential, yeah. The GABA, it's also in the brain. So these two are actually in the brain. This is excitatory, this is inhibitory. The GABA, yeah. Neurotransmitter removal from the synaptic cleft. As we said, the removal of the synaptic cleft can happen or should happen by enzymes present in the ECF. It's just waiting there for the acetylcholine to come, and then the acetylcholinesterase gonna start working. The diffuse away from the synaptic cleft تبعد أكثر وأكثر and taken back into the axon terminal actively. So recycled using ATP.